Have you ever really thought about what happens when a cell in your body dies? You know, it's not always this quiet, dignified exit. Sometimes it's messy, it's chaotic. Sometimes it's a full-on microscopic crime scene. So today, we're going to grab our magnifying glasses and we're going to step right into that world to investigate. So, what does a cellular crime scene actually look like? Well, to answer that, we have to understand the process that leaves all the clues behind, a really destructive process called necrosis. Okay, so necrosis. This is not some kind of neat, programmed cell death. Oh no, this is the cell literally bursting at the seams. Its membrane breaks, all of its insides spill out, and that triggers this huge inflammatory alarm in all the neighboring cells. It's totally unplanned, it's violent, and man, does it leave a ton of evidence. All right, to solve any case, you've got to understand the method, right? The howdunit. So how exactly does a cell get taken down like this? Well, the mechanism of demolition is surprisingly direct. It really just boils down to a 1-2 punch. First, you have protein denaturation. All the essential proteins inside the cell, they just lose their shape and completely stop working. It's like all the key machinery just grinds to a halt. Then comes the second hit, enzymatic digestion. The cell's own digestive enzymes, which are usually locked away, get unleashed, and they literally start to digest the cell from the inside out. A total self-destruct sequence. So now that we know how it happens, we can start looking for the body. It's time to perform a little cellular autopsy and analyze all the evidence that's been left behind. The clues, they're found in two main areas. You've got the cytoplasm, the main body of the cell, where we see things like increased pinkness when you stain it, and little holes start to form. But honestly, the most dramatic evidence, the real smoking gun, is found in the cell's command center, the nucleus. So let's focus right there. So the first nuclear clue is something called karyolysis. This is where the DNA inside the nucleus starts to break down, and the whole structure just begins to fade away. Think of it like crucial evidence, written in ink that's just dissolving in water. The information is literally being lost before your eyes. Now, if the nucleus doesn't fade, it might do the complete opposite. In pycnosis, the nucleus shrinks and condenses down into this tiny, tight, dark little ball of genetic material. It's like it's compacting all the evidence into one single dense spot. And that shrinking process often leads to this final, pretty explosive stage, karyorrhexis. That little shrunken, dense nucleus, it shatters. It just breaks apart into multiple fragments. At this point, the command center is totally destroyed. Case closed on this cell's life. Now, here's where it gets really interesting. Necrosis isn't just one type of crime. Oh no, there are different methods, different signatures left behind by different culprits. So now it's time for us to profile the 5 main culprits, or types of necrosis. Our first and by far most common culprit is coagulative necrosis. The classic cause here is ischemia, which is just a sudden cutoff of blood supply. You see this all the time in solid organs like the heart during a heart attack or in the kidneys. And this type leads a very specific look. The basic outlines of the cells are actually preserved, but all the internal details are just gone. Pathologists have a great name for this, the tombstone appearance. You can literally see these ghostly outlines of cells that are no longer alive. It's pretty eerie. Next up, we've got liquefactive necrosis. This is what happens when those digestive enzymes we talked about just completely win the battle. It's really characteristic of cell death in the brain, like after stroke or in abscesses that are caused by nasty infections. So the result here is that instead of a firm tombstone, the tissue literally transforms into this liquid, gooey mass. In the brain, this can actually leave a fluid-filled cavity where brain tissue used to be. It just melts away. All right, case file number 3. This one has a very famous accomplice, tuberculosis. Caseous necrosis is considered the absolute hallmark of a TB infection, and actually the name itself is a dead giveaway to what it looks like. Caseous literally means cheese-like. I know, it's gross, but it's super memorable. The dead tissue has this white, soft, kind of crumbly texture, and when you look under the microscope, all the cell structure is just gone, replaced by this granular debris. Our fourth type specifically targets fat tissue. Fat necrosis can happen from physical trauma, sure, but it's classically seen in something called acute pancreatitis. That's when these powerful pancreatic enzymes leak out and just start digesting all the fat around them. And this whole process triggers a really unique chemical reaction. The fatty acids that are released combine with calcium in the tissue and form soap. Yeah, literally soap, through a process called saponification. This leaves behind these very distinctive, chalky white deposits that a surgeon can actually see and feel. Okay, our final case takes us into the blood vessels. Fibrinoid necrosis is special because it's usually caused by the body's own immune system attacking the vessel walls. It's a condition known as vasculitis, a real inside job. The evidence here is something you can only see under the microscope. Plasma proteins from the blood leak into the damaged vessel wall, and these proteins just love to soak up a pink dye called eosin. This creates this really distinctive, bright pink staining pattern that makes the whole vessel wall look thick, messy, and damaged. So there you have it. We've investigated the mechanisms, we've profiled the 5 main culprits. But what's the big takeaway here? I mean, why does understanding this whole cellular crime scene thing even matter in the real world? Well, it matters a lot, because by recognizing these specific patterns of necrosis, doctors can make absolutely critical diagnoses. Seeing coagulative necrosis in heart tissue, that confirms a heart attack. Liquefactive necrosis in the brain, that points to a stroke. Every single type tells a story, and it points directly to a specific disease, which, of course, guides treatment and ends up saving lives. So we've established that necrosis is a violent, messy, inflammatory process. It really is the cellular equivalent of a chaotic crime scene. But that brings up a really interesting question, doesn't it? What if a cell could die cleanly, quietly? What if it could tidy up after itself without alerting all the neighbors? What would that even look like? Well, that's a whole other story for another time.